Hello, this was the sunrise on this morning that I made this video whenever you're going to watch it. Hope you enjoy it. Here we go with cell transport. So this is about how things get in and out of cells. In class we might do a lab or two, hopefully, where we use some kind of models of cells, like gummy bears can be used as models of cells. Ziploc bags can be used as a model of cells because things can go in and out of both of those, right? So we'll get back to a diagram like this real fast. Slides. So what exactly needs to get in and out of a cell, right? We mostly focus on what needs to get in a cell. Remember in the last cells unit we said all living things, cells or big multicellular things, need to get nutrients in. Nutrients like what? Well, there's nutrients like glucose, sugars, nutrients like sodium, nutrients like water. There's all kinds of nutrients, right? Both organic ones like glucose, inorganic ones like those two. And then what kind of things need to get out? Well, wastes need to get out. But sometimes so do always also things that aren't quite waste. They just need to go somewhere else, like glucose, to get out of one of your liver cells, to go into your bloodstream, to get to one of your brain cells, for instance. So a good word that we're going to use that describes all of these, whether they're ions or molecules and organic or inorganic, is particles. So that's what we're going to collectively call all these things that are going in and out of a cell. So what determines if you can get through this selectively permeable plasma membrane, or even the membranes inside the cell like the nuclear membrane? We're mostly going to focus on stuff in and out of a cell, but it's the same as in and out of a nucleus. What kind of things can pass through? Well, their size makes a difference, right? Smaller is easier to pass through, right? And the other big thing is their charge, and that's because of the nature of the plasma membrane, having those charged phospholipid heads in the phospholipid part. What does a membrane have in it that might help something get through if it's charged and it can't get past through that lipid bilayer very well? Well, that's the proteins in it. So here we go. Now that we have that kind of set up, the structure of a membrane has everything to do with its function. So we have different kinds of proteins that help in transport. So we can call them all transport proteins. Now we have, starting with a little bit of specific definitions and an idea of specificity at the same time. So here's what that means. Some kinds of proteins are called channel proteins. They're just kind of like a tube that lets things go through. But then pump proteins actually change their shape and, as their name suggests, physically pump things through in either direction, right? But this specificity word refers to the fact that for everything that goes through a cell membrane, like sodium ions, for instance, there is a specific protein that helps, if indeed it needs the help of a protein. So that's the specificity thing. For sodium to go in and out of cells requires a specific protein. For calcium to go in and out of cells requires a different protein. And their shape and their charge has everything to do with whether that protein can let something through or not let something through. Okay, so here again, here's the red ink. That means this is the EOC. This is what they might ask you about transport. This is part of it anyway, right? So we have here terms active and passive. These are key terms. They're not too hard, but they're a little difficult in a couple little nuances. So we are looking at how particular structures, like the structure of the membrane itself, is again related to its function. We're always looking for that. So back to these passive and active words. What you notice here in the active little diagram is ATP. That's the molecule that we've already studied that is like a little battery. And that's what the active refers to. It refers to the need for energy for this transport to occur. But every kind of transport needs energy. For these molecules to move across here, there has to be energy in the system. Things don't move without energy. The key difference here is that in active transport, it's energy from the cell in the form of ATPs that has to be used to make this transport happen. Okay. Now, some of the dynamics of transport, we'll come back to this diagram like 4 times during this slideshow, but I'll just point one out here. Passive transport, in this case, is also known as diffusion, and we have simple diffusion. They didn't put that word there, I'll put it there, which means those particles are going through the bilayer part. Or we have facilitated diffusion, where those particles are going through with the help of a protein. Facilitate means to help, so that's what somebody named this kind of diffusion. Okay, what's going to be the difference whether something can get through the bilayer or needs the help of a protein? Charge. So all these things are going to be small, smallish, but the charged ones are going to have trouble getting through the membrane because remember, these phospholipid heads are negative, actually, I'll make it that way. They're negatively charged. And so if you're a negatively charged thing and you get close to here, you're going to be repelled by it. If you're a positively charged thing, you get close to here, you're going to stick there. You're not going to go through very easily. But if you're a non-polar uncharged thing, you can go through simple diffusion-wise, you can get through the bilayer. If you're a charged thing, you're going to need the help of a protein. Like a sodium ion, for instance, you need the help of a protein. Okay, so what is this diffusion? That is not quite a synonym for passive transport, but it's going to be close enough for us. Okay, what diffusion is something that happens naturally, without any help, without anything for anyway, right? Particles are always moving, unless they're what? In a situation that we call what? Absolute zero, right? No energy in the system at all, which is a theoretical thing because you're not going to find that kind of situation on Earth, right? Even in a laboratory, we've not been able to create absolute zero situation, right? So that's why they move at all, is they have some energy. In the system has some energy. The more energy, the faster they're moving. So what diffusion simply is this. If you have a whole lot of particles in one place and they're all moving, and here is a key word here, they're moving in random directions, nothing's pushing or pulling them in any certain direction, their movement is random, then they're going to spread out. This random movement of particles from where there's more of them, more concentrated, to where there's less of them, less concentrated, is named after the guy that first described this. His name simply was Brown, Robert Brown. It's called Brownian motion. He's also the guy that named the nucleus of a cell, for what that's worth. And here we have this idea. When those particles start to move out, they're still moving at random. So if I have a particle out here, it might be moving back towards where there's more of them, right? But since there's more over here, there's going to be more moving in that direction. I'll put 3 arrows, moving from more concentrated to less, and 1 arrow moving from less concentrated to more. That's the idea of net. Just like it is in a business, you have a net profit, right? If I made $1,000, but I had to spend $300 to make that 1,000, then my net profit is 700. So likewise, if I have 3 particles that move that way, for every 1 particle that moves that way, I have a net movement of 2 particles that way. That's what diffusion is about. Net movement from an area of higher concentration to an area of lower. So now by definition, to have diffusion, you have to have those 2 areas. So here is a diagram we're going to look at about 5 times, right? Because here we certainly have the situation that diffusion requires. We have an area of high concentration of yellow particles in this case, right? We have an area of, in fact, 0 concentration right now of those particles. And this time in our little diagram, we've put a membrane in between those 2 places that's like a cell membrane. And you can see they made this one so that the little spaces in between it are big enough to where those can pass. This membrane is permeable, but it only lets things that are small enough pass through. It's selectively permeable. Okay, so now if we start the clock from this situation, you'll get what you expect. Particles are going to move in all directions, up, down, left, right. But the ones that are going to the right, if they happen to hit one of these holes, now they're on the other side. Just like these four have, right, at this point in time. So if we stop the picture at this point in time, we've had diffusion happen. But we still have a difference between left and right. And now the difference, it's not on this, so I'm going to draw it here. The difference is called a gradient. That's what word someone chose to mean difference here when we're talking about concentrations. We have a concentration gradient. More on the left, less on the right. So these particles are still going to be moving randomly. Again, might this particle bounce into the wall here and then bounce off this way and go back this way? Yes, it might. But simply since I have more of them over here, there's a greater chance that ones are going to go from the left to the right than there is from the right to the left. And again, we'll have a net movement from higher concentration toward lower. Eventually, though, they're going to all get equally spread out. Equally spread out in terms of concentration, right? Which in this case, also we can numerically count. I've got 8 particles on each side, and these are roughly the same volume. That's what concentration's about. How much stuff do you have in a certain volume, right? So now, what these particles don't do is say, we finally made it. We're equally spread out. We can rest now. No, they're still moving by Brownian motion. They're still randomly moving. And some of them is going to have their movement take them this way, but then other ones are going that way. And if they're equally spread out, it's likely that every movement in one direction is going to be canceled out by a movement in the other direction, and they'll stay at this steady state we call equilibrium. It's a dynamic equilibrium. Again, they're still moving. That's kind of what dynamic means, right? But here we have a state where there is not going to be any change. Unlike these two places where as time went by there was change, here we're not going to have any, what kind of change? Net change, right? No net change. What's that mean? Here's what it means vocab-wise. When we talked about more particles moving from more to less over here, we said they're diffusing. Now, particles are still moving, but since the definition of diffusion is net movement, and we don't have any net movement, we say in this system, there's no diffusion happening. Even though there's movement, movement and diffusion are not the same thing. Okay, a little bit of a difference, but it's a kind of a key definition difference. So now we have this idea. In that simple system, there was just like one kind of particle, right? We're going to say this word too since it's on the screen. These are particles that are dissolved in water. We'll get back to the water in a minute. Those are called solutes, right? Water's called a solvent. We'll come back to that again in a minute, right? But there's a whole lot of particles in a cell. They're dissolved. They might not even be dissolved, but they're moving around at Brownian motion, and they all have their own separate gradient. So here, these two particles, the dark ones and the yellow ones, they have their own separate gradient. More dark ones over here, less over here. Yellow ones, more yellow ones over here, less over here. They're all randomly moving around, and they're going to diffuse down their concentration gradient. That's how we would say it. The purple ones are going to have more go from left to right. The yellow ones more go from right to left. And after a little while, this is what it'll look like, right? But we still have a gradient for both of them. We still have a higher concentration of dark ones on the left and a higher concentration of yellow ones on the right. And so they're going to continue to move that way until, again, they're both now going to reach an equilibrium. So far, so good. Point is, every kind of particle has its own gradient. So the direction of diffusion of one might be different than the direction of diffusion of another, and will be. Okay, so here we've already said this, right? If you're a charged particle, like sodium, and you can't get through the lipid part of the bilayer, you may well have a protein, like this, in that bilayer that facilitates your diffusion in and out. But it's still always from where there's a higher concentration of you to where there's a lower. So we go back to this again, right? So here's the protein that we had this word before. We would call this kind of channel. All right, we would call that kind of channel. We would call this kind of pump. We're not quite ready to talk about them yet, but we're getting closer. So that's the difference between proteins. Are they passive transport proteins that help facilitate diffusion? Call them a channel. So sodium channels, for instance. Are they a pump protein that actively, with the help of ATP energy from the cell, moves these particles? We call them a pump.